Hi, this is Dr. Laila Al-Haddad from Kuwait University Chemistry Department. I'm very glad to share in the International Conference on Cardiology and Cardiovascular Medicine under the Best Researcher Award category. My project under the title Sulfonamide Derivatives Mediated Breast and Lung Cancer Cell Line Killing Through Tubulin Inhibition. So I would like to highlight that I'm focusing on tubulin because it plays essential roles in cell signaling, division, proliferation, and other cellular functions. Therefore, tubulin is a potential target for anti-cancer agents. Clinically, the classical tubulin inhibitors have a low therapeutic index, multi-drug resistance, and induced tubulin gene mutations. Therefore, the discovery of alternative tubulin inhibitors is critically important for cancer therapy. So the goal of this study is to synthesize new sulfonamide derivatives to combat breast and lung cancer by tubulin inhibition mediated mechanism. What we did is sulfonamide derivatives synthesized and fully characterized chemically and physically in terms of melting point, magnetic measurements, IR and proton and carbon 13 NMR, thermogravimetric analysis, and the molecular weight. Moreover, we run Western blot and MTTSA to measure the cytotoxicity of our agent with MCF7 breast cancer cell line and A549 lung cancer cell lines as a surrogate models. So if we focused on the left side, you can see that alpha beta tubulin combine to form protofilament. This 13 protofilament will form microtubule. This is important for the mitosis process in the for the cell division. So if you can see the right side of the picture, you can see that when the chromosomes align into the left and right of the cell in the corners, you can see that this microtubule pull together every chromosome in each side, and we end with two identical daughter cells. Therefore, tubulin is considered an attractive molecular target for chemotherapeutic to induce cancer cell death. So we started from 2-amino-5-nitrophenol to end with this chemical structure. Position A, position B, position C will be fixed. We will not change them. But position D, we will apply structure-activity relationship and we will add 4 different substituents to end with sulfonamide derivatives L1, L2, L3, L4. This is the scheme. So we started with 2-amino-5-nitrophenol. We reacted that with 2-chloromethyl, 1-methyl, and 4-trifluoromethylbenzene in the presence of potassium carbonate and DMF. So this will give us compound A. Then compound A will react with the methyl sulfonyl chloride in the presence of sodium hydride and DMF to give us compound B. Compound B, this hydrogen will be methylated under the presence of sodium hydride and DMF. Okay, when we receive compound C, we will reduce the nitro group to NH2 in the presence of iron chloride, zinc, DMF, and water to end with compound D. Compound D in the presence of 1,4-dioxane, potassium carbonate, and the R group. The R group, we used 4 compounds. First one, 4-methoxy-benzoyl chloride, which give us L1. Okay, and then second one, we used 3,4-dimethoxybenzoyl chloride that obtained compound L2, and we used number 3, 4 iodobenzoyl chloride, which give us compound L3, and we end with using 2-naphthyl chloride to obtain compound L4. Okay, so we have L1, L2, L3, L4, four compounds similar in the right side and the middle side. The moieties kept fixed. We change only the left side, the R group. Okay, so after that we collected the data, the molecular weight, the color, the melting point, and the percentage composition of carbon, hydrogen, nitrogen, and sulfur in each compound, L1, L2, L3, L4. And further, we confirm the existence of the compound by using mass spec, and you can see in each circle, the red circle, that we confirm the molecular weight of L1, L2, L3, L4. We continue characterizing the agents using IR spectrum to make sure that all the functional groups peaks is existing there for L1, L2, L3, and L4. After that, for each sulfonamide derivative, we run proton NMR and carbon 13 NMR. So this is for L1, L2, L3, L4. Further, we did thermogravimetric data. So we used high temperature to know at which temperature our compounds or the sulfonamide derivative will be degraded or break down. So this is the graphs for L1, L2, L3, L4, and the data summarized in this table. You can see it. And so first, when we synthesize the compounds, and then we make sure by the characterization that our compounds are ready. We wanted to test them on two different cell lines. MCF7 is a breast cancer cell line, and A549 is a lung cancer cell line. So we grow the cells in the plates, and we collect them and lysis the cell and run Western blot to collect the proteins. And you can see from the first bands that tubulin exists in these two cell lines. Okay, so beta actin, we use this as a housekeeping or the control gene. After we knew that these cell lines produces tubulin, we wanted to target them. So we treated these two cell lines, MCF7 and A549, with the four sulfonamide derivatives, L1, L2, L3, L4, and we found that our agents are potent in both cell lines. And we found that special L3 is potent and in both cell lines below 1.8 micromolar. Okay, these runs is average of 3 runs. Okay, and you can see from this graphs that how the agents reducing cell proliferation. And generally, this schematic representation will show you our idea how this chemical agent, the sulfonamide derivative, will enter the cell and target tubulin function, the microtubule, which is stopping the cell from growing. And Western and MTTSA proved that by the results. So, tubulin is important protein and is conserved in all eukaryotic cells. Okay, and several studies showed that tubulin is important for cell division and mitosis. And if we control that, we can control the cancer cell growth. Okay. So here I can conclude that in this present study, sulfonamide derivatives were designed, synthesized, characterized, and biologically evaluated against breast and lung cancer cell lines. The incorporation of sulfonamide moiety is a helpful strategy to obtain a new excellent anti-cancer agents targeting tubulin. The all synthesized compounds are potent in reducing cancer cell growth, but the special one is the 4-iodo-benzamide sulfonamide derivative induced a potent anti-proliferation effect. Okay, in both MCF7 and A549 cell line at the micromolar concentration below 1.8 micromolar IC50 value. So the sulfonamide derivative mediated their cytotoxic effect by targeting tubulin. They are promising agent for the development of alternative microtubule inhibitors as anti-cancer agents. I would like to acknowledge Kuwait University Research Sector, a grant number SC1418, which supported this project. Also, I would like to thank RSPU unit in Kuwait University to provide us with all the facilities to do the chemical characterization and synthesis for these sulfonamide derivatives. In addition, we are grateful to Lerner Research Institute in Cleveland Clinic in Ohio, the USA, for providing the facilities to perform the biological tests. And I will never forget my colleagues in the lab, Mayada and Mustafa Sharif. Dr. Mayada and Mustafa Sharif from Kuwait University, and my colleagues and our collaborative in Saudi Arabia, both Professor Jamal Eddin Harisa and Professor Faris Al-Anizi. And we would like also to thank Dr. Professor Sadashifa Karnik and Lerner Research Institute in USA for providing us with a lab and all the abilities to use the labs for biological tests. So I would like to thank you for nominating me for the Best Researcher Award, and you can see I provide the full paper and the link, and if you have any question, you can find my email in the paper, and I will be very happy to share with you. Thanks.